Susan, I'd like you to take us back to the circumstance that led to Sonnet's decision to pursue upstream process intensification. Set the scene for us. What did things look like, and what was sort of the motivation for looking at a process intensification solution?
So the first molecule we went to development with actually was a reasonable producer. Mm-hmm. But the sensitivity to being in a bioreactor for 14 to 15 days was causing us to lose 50% of that intact molecule because of the product quality degradation that would occur when the product itself was sitting in the bioreactor too long. So the second molecule, which was right behind it, and in fact we had the learning of the first one to cut to the chase on the second one, and that was that it was a fairly low producer because it's a bispecific cytokine. So we're trying to express two cytokines on in a molecular structure that significantly impacted productivity. However, the cells grew well if in an intensified environment. We could get up to 75 million cells per mil, which is pretty good going. And that provided us the balance of productivity against the picogram per cell per day productivity. So we just got the volume we needed out of the intensified process. So it served both those purposes. It dealt with low yields and product quality, both of which are problematic when you're trying to manufacture cytokines.
Yeah. So, you've mentioned on a couple of occasions sort of that problematic, problematic nature of manufacturing cytokines. Were those problems manifesting themselves in a way that, created a problem that you felt as though you just needed to address, or was it more a matter of getting better? Were there specific challenges or inefficiencies that you felt you needed to address, or was it just a matter of, look, we need to create a better way to, do this more efficiently?
We needed to create a reliable way of doing it because, and actually, ironically, the consistency of the intensified perfusion is actually extremely consistent, which is something that we were not seeing in a fed batch process easily. The control parameters are easier to manage in the intensified process. So we actually did get better consistency across the manufacturing duration with the perfusion system than we were seeing with the fed batch mode. So that was a little bit unexpected, actually, but a pleasant surprise. Yeah. I think the other thing was the host cell protein gets to be a problem when you get into high cell densities. Now you've got to maintain viability. And we were seeing host cell protein, again, sometimes these coalute with the product and they're difficult to get rid of. So, maintaining high viability duration during that manufacturing process, all the way to the 15th day, all the way to the end of the process, staying in that 98 to 100% viable cells, had a profound impact on the host cell protein levels of the starting materials on the downstream side. And that was extremely helpful as well.
Excellent. I'd like to remind our audience real quick that you're welcome to ask questions as we talk through. Susan and Parrish have agreed that no questions are off the table, so we'll take whatever questions you have. Make sure that you hit that Q&A box. Susan, who within the organization championed the idea? What was were you beating the drum, or was there a bigger team that was kind of behind the initiative?
Well, we're a virtual company, so there's only 7 of us, and there's only one drum, and I've got it. So I'm the drummer.
Yeah. And tell me about.
But it was very well supported. It was very well supported by the rest of the executive management team and the board for pursuing a commercializable process for the portfolio, because we also have a portfolio of 8 drugs. We've got 3 of them in development, all of which have great application in intensified perfusion. They're all working extremely well, the first three in intensified perfusion. So we wanted to get a platform that would be appropriately applied to the pipeline. And since all of them are cytokines, they all have a core basis of performance. They vary in terms of productivity on a PCD level, but generally they all appear to be working quite consistently in terms of manufacturing using perfusion, intensified perfusion.
Mm-hmm. Okay, let me turn to Parrish for a minute and get some perspective from the consultant's context of the consultant. Parrish, first of all, I'd like you to share just a little bit about maybe some of the types of biopharmas that you've worked with in your consulting roles, and then maybe share whether your experiences working with those companies as a consultant resonates with Susan's experience at Sonnet, if that's sort of a common motivating factor for adopting these technologies, or if you see other, big motivations for it.
First of all, I work with many different types of biotech companies, large ones and small startups, and mid-tier companies as well. And just to echo what Susan has said, many of them are trying to strategize their manufacturing strategy to be a more productive, more cost-effective, more scalable platform for their pipelines. So there are many examples in the industry of process intensification being attempted, at least in the development labs. And in a few cases, they have gone to perfusion. In fact, there are now 11 drugs on the market that are produced commercially using perfusion technology. So it's an interesting approach to increasing productivity and producing product quality more consistently. And more and more companies are evaluating the opportunity.